All right, everybody, welcome back to our last exploration of lesson 2. This finishes up this portion of this unit, so this will be the last exploration before the end of the semester. Unit 3, lesson 2, exploration 2, we're going to talk about what actually happens during cellular respiration. We're going to talk about 3 important stages and what actually goes on in each one of those stages to help your cells make the energy that they need in order to survive. All right, so highlighted a couple things. What I'm going to ask you to do is to pay careful attention to what I talk about in this video, because I'm going to go into some detail, but I'm not going to go into all the detail that you might find in other videos on the internet. There's all kinds of really cool things that take place that are fairly complicated. And so what I need you to be able to focus on is just the key things from the textbook, the key ideas, and make sure that you don't get bogged down in too many of the details. So first of all, cellular respiration is a reaction where we take the chemical energy that's stored inside of glucose. Remember, that's the glucose molecule that we made during photosynthesis. We take that glucose molecule, we break it down, and we release the ATP that's stored inside. Now, just like we did with photosynthesis, this is a picture that I want you to make sure you're very familiar with. This picture represents the entire process. And if you can talk about what's going on in this picture, then just like photosynthesis, you're about 75% of the way there. This picture provides a great snapshot of what we input for cellular respiration, what we get out of it, and the 3 stages that are involved in this process. Okay, so I'm going to briefly overview this picture, and then we'll talk about each one of these 3 stages. First of all, the first stage in this process is something called glycolysis. Glycolysis actually takes place outside of the mitochondria and can actually take place whether the rest of cellular respiration happens or not. The goal of glycolysis is to break that glucose molecule down so that it's easier to process by the mitochondria. And when we break that glucose molecule down, we break it into a substance called pyruvate. That pyruvate molecule then gets passed into the mitochondria, and for stage 2, it's broken down in something called the Krebs cycle. We'll talk about this in a second, but each pyruvate molecule contains 3 carbons, and then the final aspect of this Krebs cycle is to break apart all 3 of those carbons. Now, at the end of the Krebs cycle, we actually have completely broken down our original glucose molecule. We don't have any bonds still together from that original sugar. But what we've done is we've collected a bunch of electrons through these 2 molecules, NADH and FADH2. And just like in photosynthesis, where we have these molecules that pick up electrons, we have the same type of molecules here. In photosynthesis, we call them Uber drivers. In respiration, we can call them Uber drivers again. You can call them Lyft drivers. You can call them taxi drivers, whatever you want. It's the same type of molecule where they're looking for electrons that need to be picked up and taken to the final stage. So once we break all those bonds, we collect all the electrons, both from glycolysis and the Krebs cycle, and we take them to our final stage, which is called the electron transport chain. And sometimes this gets referred to as the ETC for short. But the electron transport chain takes all of the electrons from the first 2 stages, and it uses them to make a ton of ATP. Now, this last stage should look familiar to you, but we'll talk about that in just a little bit. All right. Throughout this textbook, there are these little checkpoints, and I would encourage you that you always do them because it gives you a great snapshot of what's going on here. This paragraph is a great description of cellular respiration in general. Like I said, if you can talk about this picture, you're about 75% of the way there. This paragraph provides you with a pretty good description of what's going on in that picture. So make sure you don't just skip over these activities because they're there for you to be successful, and in my opinion, they're pretty helpful. All right. The first stage of respiration is called glycolysis. Glycolysis is what we call an anaerobic process because it actually doesn't require oxygen. And we're going to talk about why that's very important here at the end of this exploration. But glycolysis, like we said earlier, is where we take that glucose molecule and we break it down into 2 pyruvate molecules. Here's that original glucose. And ultimately what we're doing is we're breaking it down into 2 pyruvates. Now, similar to any of the other reactions that we talked about, it takes a little bit of energy to get this process started. But once we start this process, we actually get to generate ATP. It's not very much, but it's a little bit, and that's significant. And again, we'll talk about that. We'll talk about that later. But the main goal of glycolysis is to do 2 things. First of all, we are trying to collect a little bit of energy right here, but we're really just trying to break that glucose down into 2 smaller molecules that are easier to process. All right, so that's glycolysis. The second stage is called the Krebs cycle. Now, as with most of the things that have weird names, this cycle is named because of a guy named Hans Krebs who discovered it. Now, as your textbook says, the function of the Krebs cycle is to complete the breakdown of glucose that was started in glycolysis. Again, we're trying to take those 3 carbon pyruvate molecules right here, and we're trying to break apart the remaining bonds so that there are no existing carbon-to-carbon bonds. Now, at the end of this, again, all of the carbon is going to be broken down. We're going to release it as carbon dioxide. And I don't want you to get too bogged down by this picture because there's a lot of things going on here. And I don't necessarily need you to be able to describe every single step of the Krebs cycle. All I really need you to know is this last part. The whole point of the Krebs cycle is to complete the breakdown of glucose and collect all of the electrons that were holding them together. All those carbon bonds have electrons holding them together, and it's important that we break those apart so that we can collect the electrons that are trapped inside and ultimately take them to the last stage of this process. Now, there's another little check-in right here. Highly recommend you don't skip over this. Make sure you go through here, select the correct answer, and then go back and use this as a way to practice telling the story of this second stage, okay? All right. Again, the Krebs cycle is where we're going to take all of the carbon atoms and we're eventually going to release them as carbon dioxide because they're going to get broken apart one by one. And once again, the main point of this is to collect all of those electrons using these 2 molecules, NADH and FADH2. Again, those are the Uber drivers or the Lyft drivers or the taxi drivers or however you want to think about it, 4-hour high energy electrons to pick them up and take them to our final stage, which is the electron transport chain. All right, so everything's kind of been building up to this. We've broken down glucose during glycolysis, we've broken down the rest of the carbon molecules during the Krebs cycle, and we've collected a ton of electrons in the process by NADH and FADH2. Now, when we take those electrons to the final stage, here's what we're doing. Now, if this picture looks familiar, it should. This picture should look very familiar to the diagram that I showed you in the first stage of photosynthesis. The electron transport chain has a lot of similarities to the light-dependent reactions of photosynthesis. And I'm not going to go back there right now, but if you want to go back and look at the light-dependent reaction of photosynthesis, you will see a lot of the same ideas. Now, instead of using light energy to break apart a water molecule, we've already got the electrons that we need. The electrons from the first 2 stages are going to be used and they're going to be pushed across this membrane, just like in photosynthesis, to force hydrogen ions up into this space. Because once again, what we want to create is a high concentration of hydrogen atoms in this upper area, the inside of that membrane. Because just like in photosynthesis, when there is a high concentration, those molecules are trying to get out of there to where there's a low concentration. That goes back to that idea of a concentration gradient. And just like in photosynthesis, there is a protein called ATP synthase. That's this red protein right here. And as these hydrogen ions force their way out, they force their way out through this protein. And every time they do, it creates enough of a spark to turn ADP into ATP. Now, when this happens, again, as that electron passes through, ultimately, this reaction has to happen in the presence of oxygen. So the oxygen that's there gets matched up with the hydrogen ion that passes through that ATP synthase, and it bonds to form water. Now, this is a very important reaction that takes place. Obviously, the main point of cellular respiration is to create energy, but When we do this, we create heat. And when that heat is not kept in check, it can cause some serious problems. Now, the fact that your cells also create water is hugely important because, as we talked about earlier, water has a high specific heat and can store a lot of energy without raising its temperature. So the fact that your cells are able to create water in the midst of this process that also creates energy is what keeps them from overheating. It's basically like the radiator of your car. Without the radiator of your car, all of the explosions that are created in your engine would cause your car to blow up. But the radiator keeps it cool because it absorbs all of that heat. That's the same thing going on here. The water that is created in this process helps to absorb the heat and keep your cells from overheating. All right. Once again, here are the steps in this process. You need to learn to tell the story. These are the steps that you can go through to practice talking about what happens during this third stage, the electron transport chain, and how we use these high energy electrons to create a massive amount of ATP for our cells. All right, so this is the process of cellular respiration as long as there is plenty of oxygen present. However, there's not always oxygen in your cells for this to take place. Sometimes you might have just finished exercising. Sometimes you might have just finished, running somewhere. You're out of breath because your cells have been requiring energy, but the oxygen that they need to make a lot of that energy is gone. Well, what do you do then? Luckily, your cells have another option. If it wasn't for this process called fermentation, when your cells ran out of oxygen, you might just fall over dead. And at the very least, you would pass out because you wouldn't be able to have the energy that you need to keep going. Now, when your body runs out of oxygen, it's not a good situation. There are consequences to continuing to demand energy from your cells, but you can do this for a short period of time, at least long enough to slow down and allow yourself to recover. And that happens through a process called fermentation. Now, fermentation takes place in a lot of different ways, but we're specifically going to focus on what happens in our cells. All right, so go through a couple of these highlighted portions. The production of ATP without oxygen can happen through what's called fermentation. Fermentation is an anaerobic process, which means it doesn't require oxygen. What fermentation does is it allows glycolysis to continue because in order for your cells to be able to keep breaking apart that initial sugar molecule to make a small amount of ATP, there has to be an Uber driver per se. There has to be an NAD molecule there ready to pick up that electron that's formed. Otherwise, you just have a bunch of electrons flying around everywhere, and that would cause problems. So, again, the role of fermentation is simply to allow you to continue to pick up the electrons using NAD+. Now, the way your cells do that is through something called lactic acid fermentation. There's other types of fermentation that you might have heard of, like, for example, the type of fermentation that's used to create alcohol, but lactic acid fermentation is what happens in your cells, and it's a good thing too. Otherwise, if you were ever exercising and your body started to make alcohol, that wouldn't help you keep going very well. So, lactic acid fermentation basically takes pyruvate and it turns it into lactic acid. And when it does that, it frees up the NAD to go back and pick up another electron. The problem is, once that lactic acid is formed, it doesn't have anywhere to go. It just builds up in your muscles. And that's what creates that burning feeling in your muscles. Maybe when you're sprinting or when you're running and you can feel your muscles start to burn, it's because you're out of oxygen and your body is telling you, hey, we can't keep this up for much longer. You need to stop. Calm down, allow your oxygen to resume so that we can go back to regular cellular respiration. Otherwise, you're just going to keep burning this up, and it's kind of like driving your car at like a red line speed. Eventually, it's going to shut down. Now, once you stop exercising, the lactic acid can be broken down, and you might only be feeling the effects of that for a day or two. But if you've ever been sore the day after a workout, you felt the effects of having lactic acid build up in your muscles. Now, that's the end of respiration. My last thought is that you probably want to go back and watch this video a couple times. Focus on those key diagrams. Focus on these portions in your textbook where you can fill in a couple of blanks, because those are what's going to help you ultimately tell the story of this process and describe how we're able to create massive amounts of energy from that original glucose molecule.